Hello, my name is Craig and today I'm going to briefly go over the anatomy of the nephron, which is located in the kidneys and the basis of solute and water transport. Here is a crude drawing of the nephron, which is composed of the afferent and efferent arterioles. In the glomerulus in red, the proximal convoluted tubule, which is where most of the reabsorption occurs for water and solutes, including sodium, potassium, and chloride in green. The descending thin limb of the loop of Henle, which is highly permeable to water, which is in blue, and impermeable to sodium chloride and urea. The ascending thin limb, which has an opposite nature to the descending thin limb, since sodium chloride and urea are permeable and water is not. The thick ascending limb and distal convoluted tubule, collectively known as the diluting segment, which we will get to. And last but not least, the collecting duct, where urea is recycled back into the loop of Henle and some is left in the interstitial space and contributes to the concentration gradient. We can see on the left from the cortex, the osmolarity of the extracellular fluid surrounding the nephron increases as we go deeper into the medulla toward the nephron loop. Now, one of the functions of the kidney is to reabsorb water so our body does not dehydrate and we do not need to drink water constantly. Our kidneys concentrate urine through a mechanism called counter-current multiplication in the loop of Henle, which is dependent on the permeability of the nephron. Starting at the proximal convoluted tubule, the osmolarity of the fluid inside is about 300 milliosmoles per liter. Moving down to the descending thin limb, water is moving out of the nephrons, so the fluid has a higher osmolarity or is more salty. With the very high concentration of solutes inside the nephron, more salts are pumped out into the interstitial space, which is in the thick ascending limb. This results in a very hypotonic fluid inside the distal convoluted tubule. So we see when water is moving out of the descending thin limb, the interstitium is being watered down, so salts from the thick ascending limb are being drawn to concentrate the fluid outside of the nephron. The rest of the urine is concentrating is dependent on a hormone called antidiuretic hormone, or ADH. If we are dehydrated and ADH is present, aquaporins, which are channels that transport water, are generated and transported to the membrane to allow for water to escape the nephron. This results in a highly concentrated urine since your body is trying to not lose water. Now, if you are hydrated, ADH would not be present and water would be trapped in the collecting duct. This would result in a less concentrated urine. Now, I want to switch gears since we talked about countercurrent multiplication and urea cycling, but there is one more mechanism the concentration gradient is maintained, and that is through countercurrent exchange. With this, the loop of Henle, here is the picture of a loop of Henle and is a simplified version of the vasa recta, which surrounds the loop of Henle, which is highly permeable to water and solutes. Due to the high permeability, the vasa recta is nearly isotonic to the surrounding fluid. The vasa recta has descending and ascending component, which flows in the reverse direction of the nephron. So we see the nephron flows from left to right, however, the vasa recta flows from right, starting from the efferent arteriole to left, which leads to the vein, or more specifically, the arcuate vein. We already know how water and solutes flow at the various sections of the nephron. So for the descending thin loop, water moves out, and for the thick ascending loop, solutes including sodium, chloride, and potassium move out also. The solutes moving out of the thick ascending limb would be picked up by the vasa recta, and water would be leaving too. This results in the blood being very salty but still isotonic to the surrounding fluid. On the ascending portion of the vasa recta, the water that has escaped from the descending vasa recta would return back into the blood, and the solutes would move out of the blood vessel and back into the interstitium. Then, the blood would move towards the vein and reach the heart at a later time point. So we see the solutes in water are moving from nephron, interstitium, and vasa recta and back to interstitium in this cycle, which generates the concentration gradient in the medulla. This concludes this brief overview of the anatomy of the kidney and how water and solutes are transported. Thank you.